Good morning, everyone, and welcome to this program. This is an introduction to a program that is starting today called Bioinformatics for Infectious Diseases. And this program is going to be hosted at the University of Ghana. But in general, we'll talk about how bioinformatics can be used in the context of infectious diseases and the host-pathogen interaction. So those of you that have not yet registered to the program, you'll see an overview of how you can complete your registration, but you will also be able to learn about general application of bioinformatics to the study of viruses, and other microorganisms that cause human disease. And we're excited that all of you are joining. Hopefully, I will be able to answer most of your questions. And if not, I will also point you to a number of resources that we have available for all of you that are joining today. So let's start by talking about briefly this program. Bioinformatics for Infectious Disease is a program that is specifically designed for those interested in conducting research using bioinformatics tools. So we'll talk about where to find data, how to analyze such data, what to do with your analysis results in terms of biological interpretation. And our main objective here is to provide a support system for those of you interested in leveraging bioinformatics analysis tools to really get to a place where you can get a publication or maybe a poster presentation, and you're making progress in terms of research outcomes. So that's the main objective. What we'll do today is we'll review all of these different resources that will be included. We'll also take a look at a number of different sessions that are going to be coming up so that you're aware of the schedule and how things are going to be organized and what's the expectation, what are some of the dates. And hopefully we'll be able to answer any questions that you have, both about the program itself and about the outcomes, how to get to those outcomes that we'll talk about. Now, I'm going to be guiding this particular session, but I want to mention that this program, first of all, the oversight and the development of the resources and tools are going to be overseen by the University of Haifa Tauber Bioinformatics Research Center. So this research center is a research center focused on bioinformatics tools and algorithms. They specialize in developing novel algorithms that we will be reviewing and using during the sessions that we have. It is guided by Dr. Leonid Brodsky and Dr. Alfred Tauber, both of them with extensive research experience and collaborations. But also we have a dedicated team that is supporting our training activities, helping with all of the technical resources that we have, and maintain a whole community of thousands of users from around the world that are learning bioinformatics and applying those to a variety of different topics. So at the end of this session, you will be able to learn about some of the additional programs that we are launching in February that include programs on metagenomics, genomics, different types of applications of those as well, and in different locations, for example, in Nigeria, in Louisiana, in the US, and in India. So how do these sessions work? I'm sure that most of you are already familiar with how we kind of operate. We have a number of kind of resources that work together to make these sessions effective. First of all, we have our interactive sessions, which is something that you will experience today. Hopefully it is interactive in the sense that it's not just going to be me speaking, but we'll also hear from some of you and learn about your background and your interests. But also we have a number of guided materials that you can go through to learn about different types of data, different tools for analysis, and how to conduct a research study using bioinformatics analysis tools. And so we'll review a little bit of how these guided materials are organized and how we can make those into a part of a session. In other words, we will show you how the activity of your progress through those asynchronous resources is tracked, and how do we use it to make sure that we address the topics that everybody has questions about or is struggling with. The final one are the practical assignments. So these practical assignments are really an introduction to research tools. And bioinformatics is a comprehensive name of a whole field of research where we use databases, analytical tools, visualization of data, interpretation of data, right? So there are many components to bioinformatics. So these practical assignments are designed to introduce you to some cloud-based big data analysis tools, but also some practical skills, how to manage your data, how to effectively visualize, annotate, and work with your data. So it's going to include some coding, some big data analysis tools, and hopefully I'll clarify this in just a few more minutes. Now, the sessions are guided by a number of people. And so these are your main contacts. I'm going to introduce all of us now so that you're familiar with our team. And then I'll ask you a few questions about you so that we can also get to know you. So first of all, you probably are already familiar with Biswas. She's our community manager and responsible for logistics and accounts, and you'll be getting a lot of emails from her. So as this program starts, it's important for you to make sure that you have her email address saved in your contacts, which is marketing@pine.bio. This is an important contact because she will be sending you a lot of information about recordings of the sessions that we complete, information about upcoming sessions, as well as any assignments and additional resources that you should have access to. And so if you have any questions about your login, about your account, about how to use something, she would be the person who can direct your questions to the right person or answer them herself. The second person on our team is Dr. Harpreet Kaur. She has a PhD in bioinformatics and specialized in genomics, specifically in cancer, but she is very well versed in all of the methods of analysis that we have on the T-Bioinfo platform. So for any technical assistance, questions about methods, how to run those methods, issues with your input data or output data, please also add her into your contacts. Her email is support@pine.bio. Thank you, Bipsa. You can find those email addresses in the chat box. Dr. Mohit Mazumdar is our project mentor. He will help you work with available data on public domain repositories, identify that data, make sure that you understand how that data has been generated, and help you translate your idea about how to research that particular data set into a research question. So you can also find him on his email, which is mohit@pine.bio. So he will be helping you and guiding you for the different projects that you have already maybe thought of, or you're starting to think about and develop throughout this program. My name is Ilya Brodsky. So I'm going to be helping by conducting some of the live sessions and also helping you by linking you to the right resources that we have on our platforms, on the different websites that we operate. And also I could help by providing some feedback on the research projects as they develop, or any other technical questions that you might have. A lot of times I will leverage our collaborators at the Tauber Bioinformatics Research Center and getting you good information from them to answer any scientific questions that you might have. So let me tell you a little bit more about our team, what is their background, and what do they really specialize in. Dr. Mohit Mazumdar has over 10 years of experience. He's got his PhD in bioinformatics or chemoinformatics primarily. He now works with a number of faculty and university contacts, as well as industry contacts that we have. But his PhD was focused on developing a method to identify and predict calcium binding sites and study binding affinity with calcium. So he had to really master data mining and become proficient at predictive modeling. So he would be especially helpful once you get to one of the sessions where we will discuss structures, how proteins interact with different ligands, how proteins interact with other proteins, how do we deal with structural biology questions, even though he's also familiar with a number of other applications of bioinformatics that we will cover, like analysis of NGS data. Now, as I mentioned, Dr. Harpreet Kaur, she has her PhD in focused on prognosis of liver cancer, identifying biomarkers, and primarily her expertise is with genomic and transcriptomic data. So you'll find her expertise primarily on how to analyze big data, how to apply machine learning to gene expression data, genomic data, how to integrate data through multi-omics integration methods, and how to develop a sense of what are the specific features in that data that are most insightful, that are most important for our ability to differentiate between different disease stages or disease conditions, or maybe different types of population representatives. So that is her background. Now, I told you a little bit about our team. I would like now to also ask you about your background. So I'm going to launch a poll. This poll will ask you the following questions. So first of all, sorry, 1 second. That should be later. So first of all, what country are you from? So we have a number of countries that we expected people to be from, including Ghana, Nigeria, India, USA, or maybe other. So if your answer is other, then please put in the answer into the chat box. Bipsa, do you see the poll? Is the poll live now?
Yes, Ilya, it's live now. Okay,
so please answer. The question number 1 is, which country are you from? Now, the second question is about the city. Which city are you from? We also anticipated a couple of cities. We don't know exactly where you're from, but Lagos, Kolkata, Accra in Ghana, or Delhi, or New Orleans, Boston, or maybe other. So please, you can, if you don't see, I don't see any answers right now in the poll yet, but please respond in the poll. If it's other, just click on other, and then you'll be able to put in the response there. Okay, university. So again, this program is primarily hosted by the University of Ghana. And the people from that university, we have that answer in the poll. University of Ghana, Louisiana State University, Haifa University, or other. So please put your responses again in the poll. Number 4 is about your degree. So your degree could be bachelor's, master's, PhD, or other. And then another couple of questions. Research interest. So your interest could be in virology, microbiology, genomics, bioinformatics, or other. And then what is your background? Your background could be researcher. So the background could be researcher, student, faculty, industry, or other. So again, this is a poll. You should be able to see the poll. And so please add your information from answering the questions in the poll. And I'll give you a couple of moments here to answer all of these questions. I know that it's a lot, but it's a good way to get to know everyone and really understand our audience a little bit better. Okay, great. So what we're seeing is we have about over 50% of the participants here are currently researchers, and so that's great to know. About 7 to 10% are faculty, so that's great. Now, I'll leave this poll on for now, but I'll continue with my slides. And the primary thing that I wanted to get to is that we have this free course called Introduction to Bioinformatics. And the reason why we start with this course, one of the reasons, is because it is a course where we have a survey. In the survey, a lot of the same information is being asked. And thank you, Bipsa, for putting that in the chat box. So let me show you how to access this course. Please go with me to edu.t-bio.info. So this is the link. Okay, thank you, Bipsa. And let me know once you're there. Okay, you can put yes, or I'm there, or number 1. And yes, just again about the poll answers, if you could please use the poll to answer the question, so that way we'll have a structured data set at the end and we can review the data together with our collaborators. Thank you, Bright. Okay, thank you. So if you're here, this is a website where we store a lot of our asynchronous materials, including some of the important information for this program itself. So if you don't have an account already, you need to create an account. If you are from the University of Ghana and you have registered for the program through University of Ghana, your account information should have been sent out to you. So please use that information to sign in. And I'll just briefly show you. Once you're here, you can go to courses, and this is the course that I'm asking you to take a look at. It's called Introduction to Bioinformatics. And this is a course that on one hand is a course designed for beginners. Maybe you're thinking, I am already a researcher or a faculty. I know a lot about bioinformatics, and we of course understand that. And we're not saying that this is going to be some revolutionary course, but it's a free resource. And one of the important things in this course is that it has an intro survey. In this intro survey, we collect information about the users, primarily to identify major trends and interests of the people that are joining our program. So as a part of this infectious disease program, you will see some of this information that we already collected in the poll, but you're welcome to submit that information again, because we're also interested in your bioinformatics experience, in your general background, why you want to learn bioinformatics, or maybe already learning bioinformatics, what would you expect to take away from these courses, and if there's anything else that you would like to share, for example, some specific type of interest that you have, and we haven't mentioned it in the survey, you can ask that, or you can answer that right there. Okay, so again, please take a look at this course, and we hope to learn more about all of the participants here. Now, the other thing that you will notice is that we have in this portal a way to really tie in all of the progress that we are going to be doing in this program into your profile. So I'm giving here an example of a profile, somebody that has previously completed our Bioinformatics for Infectious Disease program. And I want to mention that it's really an impactful part of completing your profile to make sure that your certification is up to date. In other words, when you complete a course, you will get an automatically generated certificate. That certificate could be shared on social media. It could be used for other purposes, but it's important that your full name, the way that you would like it to be on your certificate, is spelled out right here, right? So for example, this is going to be your username once you sign up, but this is going to be your full name. Now, it's also interesting for us to know if you're a faculty or you're part of a company or something like that, that you add that information right here. And we anticipate many of you to have a social media profile on Facebook, on LinkedIn, on other platforms. So please add that information there as well. And Beeps also posted a link in the chat box on how to complete your profile. So here, we would like to ask all of you to take some time, maybe after the session, add that information in there. Now, once you have completed your registration for the program, you will also see a badge. And this badge is an indication that you're a part of the program. And so for the duration of that program, this badge is going to be available. After you complete the program, you will get a certificate, and I will show you in your profile where you will see all of that information, including your certificate of course completion, your certificate of program completion, and as you prepare a project of your own, also your project is going to be added there as well. Now, once you have made your profile on the T-Bioinfo platform, we also invite you to join our social media groups. And these groups are not just for us to, share updates. This is an important place where we share different job openings. So if you're looking for a career in bioinformatics, there's a lot of positions open. There are a lot of collaborative projects as well. So you will find information about different resources, important updates about our programs, but also other programs and other resources that are out there. And we hope that you can join these programs. Again, Beeps have posted the social media links in the chat box, so you can find those there. Please do connect with us. So you will find myself, Dr. Mazumdar, Dr. Kaur, Beepsa, and everybody else that works for a company listed in those groups as well. So we hope to connect with you on social media, stay in touch, and hopefully share updates about this program and others as you join. Okay, so let's talk about bioinformatics for infectious diseases. So again, this is a program that is intended to support research projects through training. So we're going to do some training, we're going to do some exercises, and to explain how these exercises are organized in this session, I will give you a sense of what those exercises will be and how to find project examples that illustrate the impact of these different types of analysis techniques. So first of all, we'll talk about sequence variation and alignment. So you're probably familiar with methods of aligning two sequences, which is called pairwise alignment, or multiple sequences, MSA. But we'll take a look at some of the importance of different types of variants and different types of mutations that cause changes in the amino acids, maybe that cause stop codons to appear. And we'll think about how to find this important variation, especially when we deal with complex sequencing. So we'll talk about sequences of individual genes or regions, as well as full genomes. And we'll talk about the consequence of that. Now, another part of sequence alignment is the reconstruction of the evolutionary tree. So we'll talk a little bit about phylogenetic analysis and how these sequences can inform us about the molecular clock and how they could have originated from a common ancestor, and how do we piece that information together. We'll talk about how to map such genomic variation onto protein structure and explore the structure changes using 3D representation of these proteins produced by cryo-EM microscopy. We'll talk about also transcriptomic analysis of host response. So we'll take a look at how do we analyze the impact that is, for example, in the context of a disease on the host, and what are some of those genes that are differentially expressed? How do we map that onto a pathway? How do we interpret the analysis results? But before we jump into these specific topics, let's talk a little bit about infectious and non-infectious diseases. So an infection is the invasion of an organism's body tissues by disease-causing agents, their multiplication or replication, and the reaction of host tissues to the infectious agent and the toxins that they produce. So an infectious disease is also primarily known as transmissible disease or communicable disease, which is an illness resulting from an infection. So one of the major components of infectious diseases as opposed to non-infectious diseases is that, one, they are caused by a pathogen, and two, that they are shared from between multiple people or between multiple hosts. Non-infectious diseases are not caused by a pathogen, cannot be shared from one person to another, and are caused by other factors such as environment, nutritional deficiencies, lifestyle choices, or genetic inheritance. So because we are separating these bioinformatics programs into different categories, this is one of the reasons why we want to focus specifically on infectious diseases, because the topics that we will go over include different pathogens, and you can see some of the examples here like flu, HIV, malaria, and we'll talk about the impact on the host, right? And you can see that right here, for example, sickle cell disease, or we can talk about how the reaction to different toxin happens. So what are some of the important things that we will discuss in the context of the program? We'll talk about replication and or colonization. So we'll talk about how does that process happen. We'll talk about the result to the host cells, for example, cell death. How does that happen? As a result of what? And the release of toxins and how the impact of cell death and the release of toxins has an impact on organ function, interference, and damage. So interference with that normal function. Now, another important part that we'll talk about is the response, right? And so our bodies have these response mechanisms that include different ways that our body deals with these intruders. And we'll talk about how the immune system has several different types of responses. These responses can be trained or induced, and that is really the basis for many of the interventions that we are going to look at. And we're going to look at how those interventions work in different patients or in different examples. And we'll also take a look at how those responses could be induced and what are some examples of such interventions. So as you know, vaccines are one of the first types of interventions that has been introduced, and it has been introduced as far as back as the early 1800s. And so, you know, it's been a long time since the concept of vaccination has been introduced. But as you can see in this chart below, that is not the only way that disease burden has been reduced. In fact, we can see that other types of interventions, for example, we can see antibacterial penicillin. We can see here the vaccine that was introduced. And we also see other types of interventions, for example, continuous municipal use of chlorine in water in the United States, right? So in the context of the history of dealing with infectious diseases, there have been major progress and major successes. At the same time, now we know that that has not been the solution to everything that is out there. So in fact, with COVID-19, we are reminded once again of how morbid this infectious disease as a pandemic can be, even with all of the tools that we have at our disposal. So while the history of dealing with infectious diseases is full of successes that we can celebrate, we can also be reminded about the continuous need to study microorganisms and infectious diseases, develop novel ways to resolve them, but also develop technologies that help us understand how infectious diseases are causing disease and where do they come from, where do they originate from. So one of the ways that we will look at this, we will see that while in the beginning, one of the primary tools that we had to understand microorganisms was the microscope, but today we have additional types of information like genomic sequencing that really helps us address the impact of infectious diseases, understand virulence of these different pathogens, understand their protein expression, understand how to diagnose them more precisely, insights into the molecular mechanisms that they interact with the host and with the vectors, and also understand how do they evolve, how do they change from a pathogen that might not cause such a harmful effect to a pathogen that could be very deadly. So what are the processes, as well as what are the specific manifestations of those processes. So we'll take a look at these different technologies like sequencing, and we'll start seeing how do we apply those in the context of specific project examples. So one can also think about differences in viral responses. We can think about different strains. We can think about different types of responses. And a lot of that information could be captured from sequencing. But of course, sequencing could be of many different types. And the important part for us is we will take a look at how from sequence data, we can start looking at structural analysis. We can look at gene expression studies, And as the data becomes available, determine patterns that could be impactful for our detailed understanding of protein-protein interaction, as well as single cell level response to these different pathogens. And how do we piece all that data together? So to really take a look at such a diverse range of topics, we have prepared a number of different examples. And in those examples, we'll take a look at viral genomics and evolution. We will take a look at microbiome and the infection through the microbiome. We'll also take a look at different examples of immune response biomarkers to understand how this process happens. Now, before we go on, I wanted to make sure that I do pauses. As I said, the intent of these sessions is to create an interaction, right? And not only kind of a lecture. So I'm really looking forward to learning more about your interests and to understand whether some of these topics that we raise, I'm going to be pausing and asking if you have any questions. Now, as we go through and you have, again, non-technical questions, you can post them in the chat and Bipsa should be able to respond to those directly. If you do have questions about the topics that I go through in the slides, this would be the opportunity to hear what your questions might be. So again, if you have any questions right now, you can put those in the chat. And in the meantime, I am going to share the results of the poll. Bipsa, can you see the results of the poll?
Yes, Celia, I can see the results of the poll. So for the first question, which is, what country are you from? So we have 30% of attending participants who are from Ghana. 7% participants are from Nigeria. India has about 26% attendance, and we have 2% from the US as well. And 36% of participants, which is the largest number, are from other countries. And some of those countries which were mentioned in the chat box were Pakistan, Brazil, and a few other countries. So if you still want to add your country, you can, or your city or your department and name, you can again put that in the chat box if you have not answered. Going to the second question, what city are you from? So we have 26% of participants who are from Accra, 2% from Lagos, 2% from Kolkata, 5% from Delhi, and there are 64% who are from others. And which university are you currently in? So we have 28% participants today who are from the University of Ghana, and we have the rest of the participants from different universities around the world. And what is your current degree, which is a fourth question. And we have 46 percentile who are in a master's program, which is the highest number. Then we have 30% PhDs, bachelor degree holders are 20%, and 5% are others. While the research interest is the highest in bioinformatics, which is 75%, followed by genomics, which is 46%, followed by the same percentile of 26% in both virology and microbiology, while there are 23% who are interested in other research. And regarding the background, which is the last question, we have 46% who are researchers joining us today, and this is the highest number. While 38% are students, we have 10% faculty and 3% industry, while there are also 3% others. So this is the poll result from the session so far. Thank you, Ilya.
Thank you, Bipsa. I do also want to mention that I see, and maybe you could put in the chat box again, the links to social media groups. I see Dorothea Obiri from University of Ghana, Pratik Paul, student at Sam Higginbottom University, Ali Al Zayer, biomedical informatics, doesn't say from where, Ismaila Adams, data scientist and researcher, and Elionai Maura. So those are the people that have joined the group. And if you are still not there, so please join the groups there on social media. Okay, so let me address some of these questions. Ahmad Gifari is asking about whole genome sequence. It's mentioned that we can detect mutations related to virulence. How are the researchers conclude their findings from whole genome sequencing into more transmittable virus? So I think here you're really talking about association between phenotype and genotype. So using whole genome sequencing, we can identify the mutations, but using other type of information, for example, rates of transmission or virulence, and those are going to be either collected from the population, like rates of transmission, or they could be studied in the context of an experiment. So one of the things that we'll talk about in our next session is how this data is generated. And we'll see actually how one of the examples, we'll take data from the Ebola virus outbreak from 2014 to 2016. As you know, it spread in the West Africa. And it was the first example where an epidemic was also paralleled with almost real-time sequencing efforts. So there was a lot of data that was coming from different groups. And we'll see how in the context of that epidemic, it's a good case study because it started and it ended, right? It's not like the ongoing pandemic that we're still kind of looking at. So what we are going to look at is how to find those associations between mutations, and the phenotype. But the overall idea is that you can do, for example, controlled studies where you understand the mechanism, or you can find an association. And that association could be using something like genome-wide association study, where you translate your phenotypic information into numeric data, and then there are methods to find that using a regression model. Let me go to the next question. Kwame Tay. Are all infectious diseases necessarily pathogenic organisms? From my veterinary background, I know a couple of diseases such as canine venereal tumor, Tassie devil facial tumor. These are infections, but not caused by pathogens. So again, there might be exceptions. I do not come from a veterinary background and just kind of talking about the general descriptions, but I'm definitely interested to learn more, and I'll look into that. And if you have any more clarification about those examples that you mentioned, please put those in the chat. That really sounds fascinating. Okay, the other question is, how can I identify the position of different regions of the gene in sequence if no information about the exact position from reference papers? So I think that has to do with genomes that are not well annotated. So you don't have an annotation, and you want to identify how does a gene sequence fall in the context of the full genome, perhaps, right? And maybe you can clarify the question. But I think the approach would be to first create an assembled genome. So to assemble either the whole genome, which could be very difficult in some cases, or at least in contigs, right? So into portions that are fairly large. The way it's done is using De Bruijn graphs. You can find in short sequences of reads. You can find overlapping regions and connect them together into longer sequences. And so depending on the length of the read, the coverage, other information that is available, you can probably piece together a fairly good portion and then try to map your specific gene sequence onto that region. I think that would be the general approach. Okay, how to identify the viral genes in an organism? Great question. We'll talk about that. And today I'll introduce one of the examples where you can actually find a pathogen from another in infectious, in cell lines that are being used for biologics production. So let's get to that in just a couple of minutes. By the end of this program, will we be able to identify parasite genotyping sequencing? So my hope is that yes. My hope is that very soon into the program, you will be able to take gene sequencing information, so information from a sequencer, and understand what type of a parasite this is, or what type of a genome does it map to. We'll take a look at a few examples where you can map onto a database of viruses, or you can look at a database of bacteria, and so that way you will be able to identify those. Okay, I'll take the last question: how to find SNPs in exonic and intronic region. Okay, excellent question. As we all know, genes consist of exons and introns. And so if we have whole genome sequencing, which includes the full sequence, both exons and introns, we can map them onto the reference and identify those SNPs. If we only have exon sequencing or exome sequencing, then we cannot. But I think in general, you should be able to, given the right type of data. Again, thank you for all of these questions. I'll continue to make pauses like this. So if you have a question as we go through the slides, please put them in the chat and I will try to address them as soon as we get to a breaking point. All right, let's talk about next generation sequencing. Now, again, in our next session, we're going to talk a lot more about different types of sequencing and how that is used in different file formats, how those file formats could be visualized and things like that. Today, I will give you a brief introduction. We have about 15 minutes to go through the next set of slides. Then I'll pause for a few more questions, and then I will pass it on to, and then I will show you how to access different parts of the resources that we have. Okay, so one of the things about sequencing is that sequencing technology can identify what are the specific nucleotides, but the way the data is prepared could be in many different ways. And some good examples are targeted amplicon sequencing or whole genome sequencing. So there are many different library preparation techniques that essentially mean that once the genetic material, the biological material is collected, and DNA or RNA is extracted from that sample, that needs to go through several additional steps. And those steps could determine what are you in the end sequencing, and therefore how to analyze that data. So 3 major examples that we will again look at in much greater detail in our next session are targeted sequencing, when a particular gene or even a region of a gene is amplified using PCR amplification. And then once you do the reading of that sequence or the sequencing itself, you're really trying to make sure that, first of all, you remove those sequences that helped exclude those particular regions from the rest, and you are dealing with the impact of PCR amplification on the number of reads that you're getting from different regions. Then you can map it onto a reference genome, or if you know exactly which regions have been amplified, you can only map it onto those reference sequences. So we'll take a look at that. Many times this is done, for example, in the context of the current pandemic, people want to ask the question, are there particular novel mutations appearing in the spike region, right? So they will amplify only the spike protein. Or they can ask the question, in wastewater or in blood samples of patients, could we amplify just COVID-19 or SARS-CoV-2 genome? In another case, if you're looking at the microbiome, you might want to ask the question about the composition of the microbiome. You don't want to survey any particular individual representative of the microbiome, but you want to understand, how does the composition change? Does this group of bacteria become more prominent or less prominent, right? And so then you can amplify the 16S ribosomal RNA gene, and that would be an example of amplicon sequencing. For a whole DNA and RNA sequencing, you could sequence all of the data that comes from, let's say, again, a human blood sample, but you could suspect finding specific bacteria or specific viruses. And typically you are likely to find things like hepatitis virus, maybe Epstein-Barr virus, and other types of viruses. And I'll show you an example today from maybe if we have time. So those are again, important to understand, because when we find data, especially if we're dealing with public repositories, we need to go through and understand what was the method of preparing such data before we try to analyze it. Now, the exciting part is that there has been a tremendous growth of data in various repositories, both specific repositories like viral repositories and databases, specific databases around different types of diseases like malaria, or in general, publicly available data on the host response, vectors, etc. So I think it's an exciting time for those that know how to use bioinformatics, because what we need to understand is that we should be able to find data on practically any pathogen that you're interested in. Likely somebody has sequenced it, and so there should be some information. Obviously, that's not true in 100% of the cases, but in most of the times that I've interacted with groups of people learning about a particular domain, we were able to find some publicly available data, and that gives us a head start, right? So you don't have to start in a vacuum. You really have a repository where you can find a lot of information to deal with different types of data. Now, as I mentioned, one of the additional aspects of that is that we have almost real-time sequencing efforts that allow us to sample pandemics and epidemics and other types of communicable diseases almost in real time, developing a sense of what is going on. And so one of the first things to do is to identify what is the pathogen. And so here you can see that typically one of the key aspects of identification of the primary mechanism or the primary pathogen is to have a full genome. Now, it's not easy to make a full genome if you just have short read sequences available. So people typically rely on Sanger sequencing and long read sequencing to establish how do the different pieces connect together. After that is done, you are typically able to find how individual reads map onto that reference genome. So here you can see one of the first genomes that was published for SARS-CoV-2. It was essentially these little lines here represent the individual reads, and those reads were mapped onto the closest known genome. So you can see that this is beta coronavirus from Wuhan, right? And so once you have all of these short reads mapped onto some kind of a reference, you can now start asking the questions, what is the difference? And so there we'll take a look at many different types of questions that you can ask about the sequence, primarily around how is it different and what is it that what are we biologically studying in this kind of data? So one of the things that now we need to turn to are what are some of the analysis methods that are available. And again, let's go back to where we started. We talked about sequence variation, and primarily that's found through sequence alignment, but we'll see how multiple types of answers could be obtained from alignment, including what's called reconstruction of evolution. And that could mean in the context of multiple samples or even an individual sample, as we will take a look. Now, the second question that you might ask is, once you've identified a region of interest, for example, the spike protein, how could you actually interpret the impact of specific variants on binding, on maybe other structural properties of that particular protein because you understand its function? And a way to study the function is to study the host response through transcriptomic data analysis. Now, in the context of this program, we will be using the T-Bioinfo platform. And the reason why we are going to be using this platform is because it will provide you a number of tools that you can easily use without any computational infrastructure, just in your browser, for very large data sets. And so because this is a research program, all of you that join the program will have access to a personal research account. And so that means that you will be able to upload data sets with hundreds of samples, analyze those without requiring your own computational infrastructure. And so we hope that as a result of that, you'll not only learn about how this data is generated and what it can give you, but you'll actually do a lot of that practical work yourself. So let me show you, first of all, what this platform is all about. So let's go to server.tbioinfo. And please let me know once you are there by putting #1 or yes, or I'm there in the chat box. And I'll give it just a couple of minutes for everybody to get here. Bipse, if you could also share the demo account details. Okay, so those of you that are a part of the infectious disease program, you've already completed your registration. You should have gotten an email with your account. So if you do have your account, you should use that account. You should log in with your email and a password that you were given. For all the rest that have not yet registered and want to kind of try this out, you can use the demo account that Bipse is showing right here, that she has pasted in the chat box. Okay, so I see 1, 2, 3, 4, 5, 6. So let's give it a moment, maybe until we have at least 10 people there. Okay. Okay, has anyone else logged in? All right, great. We have 10 people. Excellent. So as you look around, this is the area of analysis. You will see that this table represents a number of different technologies that generate data. And so you will see next generation sequencing data, mass spectroscopy data, structural biology data. And then here you will see a number of additional sections for downstream analysis and application to a particular type of data. For example, in virology, you will see this right here. So in next generation sequencing data on this side, for those of you that have a demo account login, you will be able to follow along using some of these demo pipelines. For those of you that have a full account, you will also later be introduced to how to upload your own data or a data set from a public repository and run these pipelines. But just to kind of give you a sense of what this is, let's take a look over here under phylogenetic tree. Okay, and we'll take a look at this first example. Okay, so here in the first example, again, I'll go back and I open this drop down, right? So phylogenetic tree, you can see that the drop down opens this up, and I'm going to go to phylogenetic analysis. Okay, and then once you're in here, you'll be able to click on these buttons and essentially create a pipeline, right? And so what is a pipeline? A pipeline is something that connects multiple steps into a sequence of scripts. So each button here represents a script. And once you have created that script, it has been added to your pipeline. And this pipeline now could be executed. Okay, so this is just an example pipeline. You can see that the next step opens up after you have completed the previous step. Okay, so now I've run my pipeline, and this means that it is loading. Okay, so as you can see, these are different types of sections that deal with transcriptomic data analysis, deal with mutation variant analysis, that deal with different types of metagenomic data analysis, amplicon data analysis. And so you will see how in the context of this full program, we will use a lot of these different sections to understand how they are applied in the context of specific questions, research questions that you have. So how do you use the appropriate method for answering that question? Now, I'm not going to necessarily go into this right now, but let me give you an overview of what are some of these methods of analysis so that we understand what they do. So the first obvious one, we kind of saw that pop up there already, is alignment, right? So we take multiple assembled sequences. This is once we have FASTA files, and we compare them to each other to identify regions that are dissimilar. So here, for example, you have G's everywhere apart from this section that has T's. Now, this is at the nucleotide level. And one of the questions that you typically would ask is, what is the impact of this particular mutation on the amino acid sequence? Because that determines function, and that determines protein structure. And so we'll learn about different types of mutations and how those mutations affect this amino acid sequence, as well as the codon structure in the DNA, so that we can understand what are the specific biases or typical expected rates of mutation, for example, in viruses, in different codons. Another example is when we deal with FASTQ files. So when we deal with FASTQ files, the actual reads in the data set that we have, they actually represent multiple variations that could be common. So they could be aligned between multiple reads that have been sequenced. For example, like here, you have the reference genome, and in the reference genome, we have a T. And then in our reads, we find 1, 2, 3, 4, 5 evidences for a change in that position. Now, sometimes that change is not as clear cut. Here we have G and A, and here we have a C. So the question is, what is this substitution, right? Is this a technical error? Is this a real substitution? What is it? And sometimes we also see evidence of insertions and deletions that could be a single nucleotide or multiple nucleotides. And so really the way to deal with such data is to run what's called variant calling. And so in the variant calling analysis, what we are looking for is not differences between different assembled sequences. We are going to look for variants. And those variants are defined by a consensus between the reads that is higher than the threshold of technical variation that we expect as a result of technical issues with sequencing accuracy. So when we look at such data, we want to identify where could such variation come from. For example, selective pressures like antibacterial drugs, antiviral drugs, immune responses. We can also ask the question about what are they associated with, right? We had a question before. How do we identify a relationship or an association between genotype and phenotype? We can talk about how variants can play a role in the structure. For example, it directly affects the amino acid sequence, and maybe the chemical property of the change is changing, or maybe it is in a regulatory sequence like the promoter region, and even though it doesn't affect the exon, it doesn't affect the protein, it still affects how it is expressed, how it is translated. And we can do what's called comparative analysis. So we can ask the question, if I have all of my variants for many samples, how could I divide them up into different groups and compare them and maybe extract the differential variants that I observe in my data? So let's take a look at an example. Let's say we have replication. So we have a virus, it's a pathogen. The pathogen enters the cell. Inside the cell, it replicates. There is an immune response. Okay, so one of the things that we can ask is, if I know the sequence of the virus that entered the cell, how can I study what came out of the cell? And so when we look at this biological scenario, we can start asking the question, what are the differences in the genomic sequences? Or maybe even more interestingly, how can we find many different results as a result of many different pathogens living together in a single cell, and then we actually observe a variety as a result of the different selective pressures. And so the obvious question, if you extrapolate this biological question to many organisms across a process of time is to start reconstructing not only what are the specific sequences, but also how do they correlate to each other. How could we organize this into some kind of a biological model, essentially that is represented by the data that we collect? And the data that we collect are the genomic sequences of individual genomes of viruses, of bacteria, etc. So phylogenetic tree reconstruction, first of all, has many different methods. And two, it could be done either by looking at the molecular clock, or essentially what is the rate of mutations that are accumulating across the whole genome, or how do we maybe link it to real time? If we have real-time metadata, we could start finding associations between molecular clock, which is produced computationally, and the real observation data, which we know from collecting different samples. And so in the context of this program, what we'll look at is how are these trees generated? So you're probably familiar with examples like this. This is the this is the Ebola virus outbreak. And you can see here how it separates into multiple clades, representing evolution, adaptation to the host because it originated from a different host, common origin, how maybe there was a particular event that caused the split into different clades, and what is the process of divergence. So ultimately, you can see that only specific ones were the most successful at surviving for a longer time. And so these examples are going to be used as case studies. These case studies will introduce you to the biological kind of background, will give you a sense of what are the types of research questions that we are going to ask, and then how do we organize our bioinformatics analysis to answer those questions with the specific discovery and with validation. So where are we getting these projects? These projects are taken from the public domain. Again, they're taken from peer-reviewed publications that typically include the data. So they describe how the data was generated, why it was generated, who generated it, right? All the questions that we need to know about data. What are the methods that they used to analyze such data? And once you get a sense of how experts and people that have spent considerable time researching these data sets, you can essentially start taking either the same data or combine it with other types of data and the same methods or new methods that you have learned, and develop what's called a project of your own. And the idea here is to, first of all, focus on projects that are novel and independent, but within the context of particular examples that we have covered in these sessions. Now, some of these examples include things like, the novel coronavirus. So again, these are projects. Bipsa, if you could put the link to the projects for people to take a look at that, where we essentially take some of the data sets that were used to kind of ask the question about origin. We look at how the different strains from bats, from pangolin, from SARS-CoV-2 were compared to each other. We looked at how those helped identify most dissimilar regions and what were some of the conclusions about the origin. We also looked at how that was translated to an understanding of the 3D structure impacts, right? So how could this be an evidence, for example, for adaptation between different species, right? So essentially what we will do is we will take a look at divergence at the genomic level. We will also take a look at the structural components, right? And what is important is we'll try to piece this information together with available structures of proteins, right? So for example, you've probably all heard about the ACE2 receptor. So we'll take a look at the model of the ACE2 receptor, how it's actually a complex, and the complex includes the BO81 sodium-dependent neutral amino acid transporter, and that's where we will apply some computational magic to align known structures to each other and understand how does the virus actually bind to this complex and what does it actually do. So we'll take a look at these examples and kind of identify the important functional domains in these proteins that we all heard about and we all hear people talk about all the time. So that's one example. Obviously, we have a whole project on the evolution of Ebola virus and how such analysis could be replicated. Again, the phylogenetic tree is just one visual that people use, but there's a lot more questions that you could specifically ask. For example, what are some of the haplotypes, right? So how could we identify differences between strains that are more effective at replicating or less effective at replicating by comparing them not in terms of individual variants, but in terms of their composition, right? So here you can see more haplotype diversity. Here you can see less haplotype diversity. And in fact, we will also follow through some literature review where these types of issues were highlighted, right? So we're not proposing something completely new, but we're actually linking it back to some of the research that already validated some of these same findings. Now, importantly, we'll look at other hypotheses that were proposed during those outbreaks, right? Where people said, we're so concerned with airborne transmission, for example, right? Or a particular mutation can have such an impact or another kind of impact. Today, kind of looking back, it's much easier to take a look at this data, look at the specific mutations that have come up in the data and try to see whether that's a direction to continue research or where does that lead us. Now, another example that we'll use is in tuberculosis as an example of drug resistance. So tuberculosis has single drug resistance, multiple drug resistance, and this is a common issue in many different pathogens. So we've kind of assembled a number of examples from these different types of data sets, viral data sets, bacterial data sets, parasites like malaria. So we'll introduce you to those data sets in our next session, and we'll take a look at different sequencing technologies. So really to understand what is the data that we're dealing with. Now, what we want from you today is to start thinking about a project of your own. Now, many of you are researchers, many of you are working perhaps on your PhD or already doing some research work. So what we want to know early on is what are the specifics about your research question. Bipsa, if you could post the link to this on the forum. So again, this is primarily for those that have already completed your registration. And I'll just show you how to find this form where you can submit this information. So again, this is for those of you that have already registered. If you go to the same educational portal as we were on before, where we looked at the Introduction to Bioinformatics course, I will show you how there is a specific dedicated area for all of you that are a part of the group. So where you should go is under events, there's a list of programs. And so the program here that we're talking about is Bioinformatics for Infectious Diseases, Naguchi. So you click on here under programs, and you will find the information specifically about the program found right here. And here you will find different types of updates. So for example, you can see that Bipsa posted here a number of videos that we have done already in the free webinars related to this program. And you will also find different types of activity from different participants of the program. But what we are talking about right now is this section on the forum. So here on the forum, you can ask questions. You can ask, if there was trouble with something, you can post it here. There are some resources here. And an important part here is the project submission form. So the project submission form is essentially there to help you structure your ideas about your research topic into an organized form that we will review. And one of our mentors, either Harpreet or Mohit, will reach out to you to make sure that we address all of the different parts of this submission, of this proposal. So here you will find project title, the problem statement. What exactly are you trying to do and why? the project summary. Where was the data? What is the data? How many samples do you have? What kind of analysis are you proposing to do? Links to the raw data so we can check whether the data could be used to answer your research question, and any reference publication so we know that you've done your literature review. So the main thing here is to really help you organize your information in such a way that an external reviewer can take a look, evaluate, and provide you with some feedback on what you're trying to do. Okay, so let me briefly summarize what are the important program resources that you should be getting access to. And again, any questions about these resources, you should be asking from Bipsa. So she's the community manager. She will give you your login information if you don't have it. She will guide you to where to complete your registration, right? So if you don't know this program registration, how to finish your registration, reach out to her. It's marketing@pine.bio. If you don't know where to find a particular project that I showed, I've shown you resources from everything that's already available, so you can ask her. Again, we'll take a look at all of these different things, but if you have any questions, reach out to her. Now, as I mentioned, OmicsLogic is not just about training. It's not just theoretical training. It's just not, acquiring skills. It is really a community. And so what's important is, as you are getting started with these different resources, we're asking you to complete your profile. And when you complete your profile, you're not doing it for us, you're doing it for yourself. We want other people to be able to find you in the program list and know that this person is from this university or this lab, that's what they're doing. Maybe that's their social media profile, right? We hope that you can connect to each other, because ultimately in any bioinformatics research project, there's going to be collaboration. And a collaboration is a big part of what we are trying to help you establish here as well. There are many international researchers, students, and faculty from many different universities. So we're really hoping that you could connect. Now, the basic tools that we already talked about are the server account, where you can run big data analysis, and courses. These are practical courses. If you're not familiar with the basics, how to do alignment, what types of mutations to expect, what are variations in gene expression, those are the types of questions that are going to be covered in these courses. They will provide you also with quizzes that you can answer to know that you have understood the material, you can answer those questions. They will also provide you with assignments. Those practical assignments will most likely use the server analytical platform to show you how this analysis is done and make sure that you can submit your assignments to get feedback. On this program page, you will find the resources specifically around this group of people, this program. So here you will find things like the forum and the courses that are included there, different project examples that we have discussed, and you will also see how you can track your activity. So let me show you what that actually looks like. Okay, and thank you, Bipsa has posted the link. Okay, so here you have this menu. In this menu, you can find the courses, and these are courses that are included in this program, right? So if you have not completed the registration, you have to complete the registration first before you have access to this. So genomics, transcriptomics, metagenomics, etc. So you will find some of these courses right here. There are also a number of project examples. So these project examples are going to introduce you to some case studies, and those case studies are going to help you understand how it was done in the context of a particular research question. Now, a lot of these might not necessarily be related to viruses or bacteria or anything like that, but they will illustrate how such analysis could be used. Okay, what is inside these courses? Inside these courses, we explain the rationale and objective. Why are we learning about a particular data type, and what is the terminology and the biological context? You will see engaging activities that will help link the specific topics and concepts to what you are going to do, so that the analysis you do is based on logic. Then we're going to include a data set or a project with an explanation of where it came from and what is this data. And you will understand what are some databases, research publications, and project examples, as well as file formats. Then you will run an analysis pipeline, and that will help you establish the logic of how these different steps are connected into a single workflow. And you will be able to see some examples of outputs that you need to look through to understand what was in the data. And finally, it will provide you with a point system so that you can compare your progress to the overall progress of the group. So one of the important things that we have found is that consistency and returning to the assignments regularly helps you understand and become more proficient. So I'll just show you what that looks like. For example, you can look through some of these example profiles. I will just show you, what a profile of a typical participant would look like. And I see that somebody has already completed their profile, Jeff. So let's take a look at Jeff's profile. Sorry, Jeff, for putting you on the spot here, but hopefully others can learn from your experience already. So Jeff has joined, and you can see that he has 300 points, right? And so those points are from completing the profile, registering on the platform, and now you can see what courses he is taking. Now, I'm not going to show you everything because things like quiz results and assignments are going to be proprietary to that's his progress, but you can still kind of get a sense of what he's doing. So he's enrolled in a number of these courses, and as he continues to build up his profile, you will start seeing progress in those courses. Now, here are some of the top profiles. You can see we have a leaderboard of the top 100 participants from all over the world. You will see that they are from many different regions in the world. And really anyone can start with this process for free, essentially, and with minimal basic subscription, you can get started. So you can see here different people that have a number of points and the change in those points. And you will see all the way to the last person in this list, and that's about 14,000 points, right? So if you want to be on this list, we'll feature you once you get to this list. Once you get essentially over 10,000 points, we will feature you, but you can take a look at some of these examples and what these people have been able to do through the course of this program. Okay, so let me pause and see if there are any questions. And Beepsa, if you could please launch, or never mind, I can launch, I think, the second poll. So we have another poll, and this is on feedback. So I'm going to ask for you to be very honest. So we are looking to improve these sessions and make sure that the sessions are informative and useful for what you're trying to do. So the first question is, how would you rate this session? I will not be offended. So you can, tell me that was very poor if you did not like it. The second question is, what did you like the best about the session? So if you like how the topic was presented, or it was specifically related to infectious disease research, and if you didn't learn anything new, that's also fine. We would like to know. And then the question number 3 is, what is your expectation from the program? So if you would like to learn about new bioinformatics tools, or you want to get introduced to virology research, or gain basic understanding of bioinformatics if you're just getting started. So those are some of the questions. Please try to answer these questions in the poll itself. And I'm going to leave this on here for a few more minutes for anyone to be able to look through. And also, if you have any questions, right? So I'm here to try and answer any of the questions that you have. And after the questions, I will introduce you to one other resource that I think you will find interesting and relevant to what we're going to do, and give you an assignment for our next time.
Ilya, as the feedback is going on, we have a question here from Ahmad Ghifari regarding the pipeline. How to note down the procedure in the methods section of publication. I'm not quite sure if that refers to the pipeline.
Okay, so as I understand the question is, let's say you've used the pipeline to analyze your data and you want to report how you've generated that data. So one of the useful things, I'll just give you an example with my pipeline. I used a pipeline for mapping data from wastewater treatments to identify SARS-CoV-2. And this is a part of a publication, so I'm going to report on it. So the good thing is that, first of all, I have a reproducible workflow. So these are the individual methods. I used bowtie to align, and then pause count and names replace, and I will find a report on this pipeline right here. So each pipeline has a report. So in that report, I will see which version of the method was used, what was the input data, what was the output data, and how long did it take for this to run. And you also referenced the T-Bioinfo platform, which was developed at the Tower Bioinformatics Research Center. So here you can use that as a reference for running it on this server.
We have
okay,
another
so the question from
Francis.
Francis. Can the project be a systematic review in the field of bioinformatics? Yes. So again, a systematic review in the field of bioinformatics, I would say, is humongous. It has been around for decades. There are many different tools, databases, and applications of bioinformatics. And I would say that, you can definitely attempt an overview, a systematic review of bioinformatics, but I would rather recommend a focused approach. So if you, first of all, learn about the general applications of bioinformatics through the Introduction to Bioinformatics course, there you will see different types of methods, applications, databases, data types, etc. So once you have done that, you can also focus, for example, on bioinformatics for infectious diseases, right? And there you can do some systematic review. Right, so I think it depends on really what is your primary objective and your focus. And since you're already a registered participant in the program, I think we'll be able to develop that idea further after a few more sessions. Bipsa, do we have results on the poll?
Yes, we do have results on the poll, and I see that there are still participants who are still voting.
Okay,
but so
well, let
far,
me answer a couple of these questions. Hold on,
right.
let's just wait on until we have, how many % overall have answered?
We have 58% who have voted, so we can wait for a few more.
Okay, thanks. Okay, so let's take a look at these questions. How do I apply bioinformatics in plant breeding research? I think that's a good question. Obviously, genotyping, right? So looking at established mutations in particular genes could be very useful. But if we talk about the infectious diseases, there are many infectious diseases that are specifically for, plant breeding, right? So they are viruses that affect, for example, different plants. There are microbes that are used as fertilizers. We had a session yesterday on metagenomics, where we talk about the application of microbiome studies in different domains, including agriculture. So I think that there are a lot of applications. And one of the projects that we have, you can start looking at it right here under projects. If you go to all of the projects, or actually right here on the very top, you can see crop drought stress and resistance. So this is a transcriptomic study right here. And that transcriptomic study helps you understand differences between drought resistant and less resistant strains of potato cultivars. Okay, another question. Good afternoon. Can it be possible to the field of virology be able to grow the interest and grow the field? Yes, so this program is specifically designed for people that, are not technically maybe versed in bioinformatics. So if you're already in the field of virology, maybe Bipsa, you could post a link on how to complete the registration to this program on bioinformatics for infectious diseases. This would be a great start for you learning about bioinformatics and trying to apply it to your particular area of research.
We have another question, Ila, from Jeff. So Jeff's question is, as we are thinking of the project, is it advisable to come up with a project on clinical trials?
Well, I think the main question about clinical trials is availability of data. Can you actually get access to raw data to do the analysis? Because a lot of clinical trials will only report data after they have completed. Okay, I'm a bioinfo student, but I don't have money to afford it, but I have willingness to learn and explore. Is there any aid or free resources? Yes, first of all, there are many free resources. Please join for free, and you have access to a number of introductory courses for free. And I'll introduce you in a second here to our coding platform, where you can also practice some of the coding and see how these things are applied in the context of R and Python. But we also have an assistance program, a scholarship program intended specifically for people that have financial needs. Bipsa, if you could paste the link to our scholarship request form. Excellent, thank you. Okay, so let's take a look at the poll results if we can, Bipsa.
Yes, okay. So as for the first question, how would you rate this session? So we see that 59% of our participants have said that they found the session very informative. 22% said that they found the session excellent, while 14% gave it a good, which is a 3, and 5% says it was okay. Moving on to the second question, what did you like best about the session? 51% says that I like how the topic was presented. 27% says that the session introduced them to infectious diseases research, and 22% said that they're already familiar with the topic and the session helped to add to their knowledge. And regarding the third question, which is what is your expectation from the program, 57% of the participants want to learn about new bioinformatics tools, while the next 30% wants to learn or gain basic understanding of bioinformatics, and 14% wants to get introduced to virology research. So that's the poll, Ila.
All right, thank you very much. So thank you for the feedback. Those of you that did not like the session really did not have to come. No, I'm just joking. So hopefully next session is going to be more informative. I will try to improve. If you send me some specific recommendations or on how we can do this better, we would be very glad to look through and address your comments. So briefly, let me now actually introduce you to the last resource on the list. If you could go with me to code.omicslogic.com. So a big part of what we do is going to be based on big data analysis. And one of the challenges in big data analysis is infrastructure. Another challenge is coding proficiency to deal with data. Okay, thank you, Bipsa, for posting the link. You can also register here for free. So please let me know once you are here and you have registered. Now, for those of you that are part of the OmicsLogic Bioinformatics for Infectious Disease program at University of Ghana, you should use the exact same email address as you are using for your registration. Okay, please let me know once you have registered. And I should get here a notification if you are registering. So please go to this link and let me know once you are in. And once you're in, what you should see is this list of courses. And I'll kind of go through and give you just a very brief example of what we will be doing during the program. Okay, so let me know if you have registered. If you're already registered, just maybe put yes, I'm there in the chat box so I know that you're there. All right, thank you, Francis. Anyone else? All right, excellent. Okay, I'll wait for a couple more minutes for everybody to get here. What this is a collection of similar topics that are explained more at the script level. So you will see here simple courses like Getting Started, for example, where you can load data and visualize it and maybe read DNA sequences in R. And one of the things that we would like to ask you to do if you have completed your registration is to take a look at this course called Genomics. So some of the things that we are going to be doing in the context of this program is looking at variants and looking at structure and things like that. So I'm going to go through this brief lesson to just kind of explain how this works. You'll learn here about DNA structure, what are the mutations and things like that. You'll also learn that DNA is organized into chromosomes and what does that mean in terms of different variants and where they are in the codons. And it will be followed by a quiz. So here you'll learn some theoretical things, and you can know that this is a theoretical lesson if it has this learn tag right here. But then you want to practice, how does this actually work in R, for example? So here, when you get to this stage, you'll see how, for example, we can assign specific strings of characters to an object, right? And you can, for example, print that object. Now, sometimes you want to compare those sequences. And here we go through an explanation of pairwise alignment first. Okay, and here's an example. So how does this work? Here, you're loading a specific library and you're putting in some strings in here. And then you have to click on run, and it will actually run right here in the browser. So after you click the run button, it will provide you with the result. And you can see that you have here success. This code has been run successfully. So as you go through these exercises, they will become a little bit more and more advanced as you go through. So once you go through, you complete, you go to the next one that is a little bit more challenging. For example, let's go to the next one. Okay, here we're dealing with multiple sequences. So here we have 3 different sequences. Okay, and let's try to run this just as an example. Okay, so now instead of just the output, you also have this link right here down at the bottom, actually shows you the sequence and shows you where the difference is. Okay, so these types of activities are also connected to your profile, and you will see under your profile that these activities are going to add points to your overall progress. Okay, so the link to registration, this is the link to register to this particular coding playground, what we call coding playground. But also, if Bipsa, you could send a link to the registration for the program. Okay, so as you go through, you will then find a challenge. So here you see final assignment. Here you're given a specific situation. You have metabolism of drugs, and your goal is to identify which mutation is going to cause problems for metabolism of drugs. Okay, so here you'll see that you actually have to write your own code and figure it out, right? So in such a way, we hope that you'll be able to practice some of the code, understand how this code is applied to a particular problem statement, and then practice your coding so that you learn how to do it on your own. So again, the goal of this program is to introduce you to a lot of the areas of bioinformatics, like the tools for big data analysis, where you will run pipelines and understand how they're operating, as well as practice different types of analysis in R and in Python, including some of the things that we just looked at. And I hope that most of you will be taking the time after this session to complete your profile and make sure that we're getting started with this program ready for your research project. So this is the assignment for our next session. Complete your educational profile on the T-Bioinfo. 2, take the free course, Introduction to Bioinformatics, and fill out that survey. We want to know more about you, about your research interest. And 3, familiarize yourself with the T-Bioinfo server and the Code OmicsLogic Playground. So those are the last 2 resources that we just had. And to remind you, this program, starting today, January 29th, will go through sequence variation and alignment, evolutionary and phylogenetic analysis, functionality and protein structure, including the physicochemical properties of proteins, and transcriptomic analysis of host response. Again, thank you everyone for joining today. We went a little bit over our time, but I will pass it on to Beepsa to kind of take over and answer any technical questions. Unfortunately, I have another meeting to go to, but for those of you that liked the session, I thank you for your feedback. Those of you that did not like the session, I am looking forward to your specific feedback, what you think should be improved, and you can email me directly by emailing ilya@pine.bio, and I'm going to look forward to learning from you as well. Beepsa, over to you.
Yeah, thank you so much, Ilya. So I will quickly start by sharing my screen. So for any of you who are having problems with logging into code.omicslogic, when you go to the site for the first time, right now I am logged in, you will notice a Get Started button. You do not need to use the password that you have for edu-T-Bioinfo or the T-Bioinfo server. You need to create a new login. It will send a password reset link to your email, and from then on, you would not need any password to log into the site. It will register your email ID. So please try that post this session so that we are able to help you with this if you face any kind of problems with this login. And please note that your Edu T-Bioinfo login is separate from your T-Bioinfo login, which is the server platform. But one thing is similar for all of these 3 platforms, which is your email ID. So please make sure that you're utilizing the same email ID. The Edu T-Bioinfo, the T-Bioinfo, and the OmicsLogic code platform should have the same email ID